All right, I think we're live.
All right. Welcome, everyone. I hope you guys are all excited for today. I know I am. I've been looking forward to this topic for a few days because this is something that I find not difficult, but very exciting, very interesting. Myself, I should probably my excitement for this topic today is kind of showing, and I forgot to start the whole thing off. I am Nate Ferguson. I'm one of the co-founders of Escarpment Laboratories, located in Guelph, Ontario, Canada. And today we have Richard Preiss, who's also one of the co-founders of Escarpment, as well as our Director of Laboratory and Research. Today we have a topic I'm really excited for, if you hadn't been able to tell. We're talking about biotransformation and how it impacts flavor of our hoppy beers. Now, this is a incredibly frustrating, at some points, topic because the research is coming from several different places. There hasn't been anyone to really nail everything down and bring it all together. There's been some people who have tried, but there hasn't been that kind of simplistic, highly executable way for us to talk about this content. Now, this content is complicated. It's multivariable. Some certain portions are poorly understood. And a lot of this stuff is heavily hop dependent, which the information for these varietals is always hard to come by. I've already gone through and seen what we're going to see today, and I can say that Richard's done a very good job at bringing all this information to one place so we can really take this information and start applying it in our brews, make it a bit more easier to understand and process. Now, before we start and before I turn it over, I just want to remind everyone of a few things. Next Tuesday, it won't be Monday because we have a holiday Monday. Next Tuesday, we're going to be going through Lacto 2.0, the road to Lacto 2.0 with Is Neto. That'll be where we talk everything lactobacillus and kettle sours and things like that. And then next Thursday, we're going to be having our sit-down interview with Barncat Artisanal Ales, all about hazy IPAs. Additionally, the following week, we're going to be having our 3-day digital conference, which the details should be released either tomorrow or very soon after. With all that being said, Richard, I'm going to turn it over to you, and let's get to it.
Awesome. Thanks, Nate. All right, so we picked this topic because it's something that comes up really commonly when we're talking with brewers. They want to know, what is the best way to use their yeast to get these sort of biotransformation characteristics in their beers. And it remains kind of a mysterious topic to the point where, I've had brewers telling me, like, come up to me and say, like, is biotransformation really a thing? Like, it seems like a myth. Like, no matter what I do, I don't seem to get any different results. And so in light of, a lot of that conjecture and just sort of a lot of different perspectives on biotransformation, we thought it would be useful to put all this information into one place, at least assemble what we do collectively know, and then try to turn that into some action so that we can collectively understand this stuff a little bit better, because it does seem a little mysterious. And part of that is that really when we're talking about biotransformation, we're talking about a lot of different things. So we're going to cover, what is biotransformation? What is the definition? What are we talking about here? But beyond just going into the sort of hardcore science of it, and I will warn you ahead of time, there is some chemistry here. It's totally unavoidable when we're getting into biotransformation. We're going to have to talk about terpenes. We're going to have to talk about thiols. We're going to have to talk about breaking bonds and forming bonds and all of that fun chemical stuff, but I'm going to try to make it digestible and try to make it relevant. So we're also going to talk about which hops actually have a high capacity for biotransformation. We're going to talk about which yeasts can help unlock those flavors from the hops. And that's all going to culminate in some practical tips for you guys. So taking all of this sort of raw data, raw knowledge, and turning it into practical suggestions for the brewers. So you have a better idea of which hops to use at which times, which yeast to use at which times, so that you can be more strategic about biotransformation in your beers. So why is this important in the first place? Why do people talk about this? And this is the main reason, right? Hop-forward beers are really popular. I think it's the statistic has stood that IPAs and pale ales are something like 40 to 50% of the total craft beer market in North America. It's huge, right? These are by far the biggest segment of craft beers in terms of sales and in terms of production. So tons and tons of breweries are making these beers. On the homebrew side, the trend is very similar. People love brewing and love drinking IPAs. So it's really, really important. And the development of all of these really cool flavor forward hops has helped to drive this segment of the industry, and has helped to unlock a lot of interesting flavors that brewers are now putting into their beers that are giving consumers totally new flavor profiles. And we can see that reflected in the numbers. So the hop usage among craft breweries is on the rise. So we can see sort of on the bottom there that the rate at which craft breweries are using hops is increasing substantially, to the point where craft breweries are really the ones that are driving the increase in U.S. hop production, and really starting to drive that narrative. And the reason they're using more hops is because there has almost been an arms race in terms of how much hops you can pack into your beers, especially with the advent of the hazy New England IPAs that do support ever increasing amounts of hops, especially as dry hop additions. But there is a challenge here, and that is hops are expensive. I'm pretty sure anyone here who's had to buy Citra, probably has some complaints about the price of Citra, right? This stuff ain't cheap. But when all of your customers are asking for IPA, or if IPA is what you really want to be brewing and drinking at home, and I support that, you're going to have to pay the price for the hops, and those hops are expensive. So there's an opportunity here. Biotransformation by yeast can help brewers get more out of their expensive hops. Yeast can do these really cool things that actually unlock flavors or change flavors and allow us to get a little bit more value out of our hops. So this could be a way, if we understand it better and apply it, this could be a way for us to be saving money on our hops and using these ingredients a little bit more efficiently. So what is biotransformation? That's really the first step here, right, is defining this. And there's definitely a lot of different ways you could define it, and we're going to limit our scope to just beer. So the way that we see it is that in beer, biotransformation is the chemical conversion or release of plant-derived flavor active compounds. So whatever we're getting out of our hops, or maybe grain or spices or fruit that's being converted or released by the yeast, we're going to consider that biotransformation. But for the purpose of this talk, we're going to really focus on hop and yeast interactions and what that means for the brewer who's making hop-forward beers. So why would biotransformation happen in the first place? And there's a challenge that a lot of these microbes face in their environment, and it's that plants are these amazing factories of an incredible number of protective, functional chemicals. They make all sorts of crazy chemicals that protect them from pests, including some of our microbial friends, that you can sort of think of as like a small chemical version of thorns on a rose, right? We don't go and just, grab a rose by the stem because we're going to get pricked. And a lot of the microbes do it the same way. And they've adapted to that. Microbes figure out how to use all of these crazy chemicals or get rid of them, and they can harness energy, or they can eliminate that stressor from their environment. So I just think this is a cool example. Just saw this movie called Fantastic Fungi a few weeks ago, before everything got crazy, and they talk about basically seeding this pile of heavily contaminated soil with all this hydrocarbons with oyster mushrooms, and that this transforms into a mushroom oasis after a few weeks, and they were able to turn that toxic material into more mushrooms. And I think that's just a good example of some of these incredible things that microbes can do when they're faced with these stresses in their environments. So when we're talking about biotransformation in hops, we're really talking about just a couple of key chemical classes. So really the things that we're focusing in on here are the terpenes. And these are chemicals that come from the plants that are flavor active. Some of them might smell like coriander or lavender, citrus fruits, flowers like roses. These are commonly coming off of the hops and present in different quantities in different types of hops. We've also got the thiols, which are these really volatile sulfur-containing compounds. They're flavor active at extremely low quantities, so they can really change the flavor profile of a beer. And these have really cool aromas. Like they can smell like grapefruit, they can smell like guava, passion fruit, or black currant, depending on which of these thiols you have. And when we're talking about biotransformation, we're typically talking about release or conversion of these two classes of molecules. So we're going to talk about the terpenes first. These are the ones that are the best understood, the most studied, especially in the context of beer, although we will get into the thiols a little bit later as well. So the terpenes, what are they? So Wikipedia says terpenes are a large and diverse class of organic compounds produced by a variety of plants, particularly conifers and by some insects. They have a strong odor. They may protect the plants or deter herbivores or attract predators or parasites of those herbivores. So what these things are doing is either serving to deter some kind of detrimental entity to that plant, whether it's an insect or a herbivore, or they might be attracting something that's actually beneficial to them, like maybe a wasp that can eat the parasite or something like that. The name is derived from turpentine. Turpentine is a terpene-derived thing. So, and certainly if anyone has smelled turpentine, you know that it has a strong odor, and that's true of all of the terpenes. These are very, very aroma-active compounds, and terpenes are a key part of plant essential oils. So if you take a plant and extract the aroma compounds from it, oftentimes with a lot of plants, many of the key constituents are terpenes. It's a really, really important part of the aroma of all sorts of plants, especially the ones that we put into our food. So why are plants making terpenes? They're making these to defend against insects, microbes, herbivores, things that are going to eat them. They can attract parasites of their own pests. So really even controlling things that can then help them to not be preyed upon. They also use these for signaling and communication. So plants have actually very sophisticated ways of communicating with each other. And part of how they do that is through some of the aromatic molecules that they make. And then some of these also serve for stress protection. So some of these terpenes can actually prevent against oxidative stress and other forms of stress in the plants, which is really cool because then, humans have been able to leverage some of those terpenes because some of them even have anti-cancer qualities. So what are they? We're going to narrow this down even further because the world of terpenes itself is quite big. So we're really going to narrow things down to one class of terpenes. And when we're talking about yeast biotransformation of hops, we're mostly talking about the hop-derived monoterpene alcohols. So these are, in the world of terpenes, these are relatively small structures. They do get bigger and crazier. But these are the ones that are really important in terms of what comes out of the hops and what can be converted by the yeast. So the three that we're going to be talking about in detail are geraniol, linalool, and citronellol or beta-citronellol. And a key point here is that they can all be interconverted. So like depending on the plant or depending on the beer, you can actually have these things sort of jump around based on the chemical or biological environment. Another thing to mention is that these are also called the rose oils. The reason being that these are aroma compounds that are very common in roses. They're a key part of the rose aroma. The reason that we know so much about terpene chemistry is because replicating the scent of a rose is an incredibly valuable problem to solve in the fragrance world. It's very important for perfumery and other forms of fragrance. And that industry is, I think, just as big as beer. So they've invested a huge amount of effort into the chemistry of this stuff. And that's why we understand it so well in a lot of cases. But I think it's really cool that you have, it's not like you just have this one thing and this other thing, and these are all sort of end products of some kind of chemical reaction. You actually have this interconnected network of terpenes, and based on the environment, based on maybe the yeasts or the chemical environment, you can have them go in different directions and create different profiles. So just diving deeper, we'll talk about a few of these terpenes that we commonly see from hops in more detail. So geraniol, that's one that shows up in a lot of plant essential oils, especially flowers like roses and geraniums. Obviously, geraniol comes from geranium, also found in citronella. The cool thing about geraniol is it's up here. It can be converted into a lot of the other monoterpenes. So it is a platform for biotransformation in the sense that if you have a lot of geraniol, you can often end up with something else through either enzymes or through other chemical means. And in terms of how it tastes, we get floral notes. Obviously, this is something that's a key component of geraniums, which is a flower. Fruity notes. Almost all of these things that mimic rose or smell like a rose or make up the components of a rose also have a citrus component. Next time you're smelling a rose, like think citrus, and you're going to pick out a lot more of that. There's actually a huge amount of citrus in rose aroma, and the terpenes are doing a lot of the heavy lifting there. And something that's important here is that geraniol is known to enhance the flavor perception of beer. So if you take a pretty boring beer and you add a little bit of geraniol to it, you enhance a lot of the traits. You enhance the fruitiness and the citrus perception and the floral perception. You add more, you get even more response. You add more, you get even more response. So there is in terms of geraniol, the more that's in the beer, probably to a point, the more flavor active it is. So already on its own, geraniol can really have a big impact on the aroma of a beer. Linalool is another cool one to me. It's something that's found in a lot of different plants. So it's found in lavender, it's found in coriander, basil, citrus. Really a supporting role in a lot of those. I would say that it's very present in lavender. If you get like lavender essential oil, that typically has quite a lot of linalool in it. The name comes from something called linolay wood, but it's also found in rosewood as well. So very common thing to find in quite a wide range of plants. In terms of what it smells like or tastes like, it also has that citrus component, floral component, woody and blueberry. One thing you can try that I think is pretty mind blowing is so I said lavender smells like linalool. So does Fruit Loops cereal. So if you have access to some Fruit Loops cereal and someone in your household has some lavender essential oil, try smelling one than the other, and I think your mind will be blown at how similar those are aromatically, and that's because of the presence of linalool. It really supports those sort of fruity flavors. Citronellol is another one. It's found in a number of different plants like roses, geraniums, lemongrass, ginger, even found in juniper. We're always talking about kvike, but the type of juniper that's used in those traditional beers in Norway, you often get a citrusy note from that, and it's probably some citronellol in there that's contributing to that. And that's kind of what it tastes like. It's very citrusy, very floral, lemongrass kind of character. Smelling pure citronellol really does remind me of lemongrass. Really just a nice, pleasant, fresh aroma. And that's something that comes from the hops and that can also be produced by the yeast. So what does yeast do in these equations? We've talked about these individual characters and terpenes. What's yeast's involvement here? And it can actually do a lot of things in the terpene world. So yeast can convert geraniol to citronellol. So we can take that floral aroma and turn it into something that's closer to like a citrus lemongrass. Yeast can convert geraniol and nerol, which is another one of the terpenes, to linalool. So it can, again, take something more on the floral end and turn it into something a little bit more fruity. Yeast can also do some other conversions like linalool and nerol to alpha terpineol. It's another terpene that we don't have the time to get into. And another thing that's really cool is that, not only can yeast do these conversions between terpenes, but it can also generate esters from those terpenes. So from geraniol, you can get geranyl esters. From citronellol, you can get citronellol esters. So you can end up with something, with the right combination of enzymes, you can end up with something like geranyl acetate, which is an even more lavendery kind of aroma, or citronellol acetate, which is very citrusy. So there's a lot of neat stuff that goes on there. And just explaining how this plays out in beer, if you take a beer and spike it with geraniol and let the fermentation proceed, what you'll see is the production of these other terpenes over time. So that geraniol is converted into citronellol, nerol, and linalool, which will change the aroma profile of the beer pretty dramatically, because you're starting with something that's floral, and you're creating all of these other terpenes that collectively may provide a little bit more aromatic complexity. So I think that's really cool. And the other part of this here is, you're asking, okay, yeast can do this really cool reaction, but what yeast is doing it? And it's really hard to find information on this. It's been hard to find studies comparing yeasts and sort of measuring the terpene conversion of those yeasts. And a lot of this information has been, especially within craft beer, has been anecdotal. We know that some yeasts are reportedly better at terpene conversion than others, but the published studies are pretty limited. We do know that some brewers are blending in Weizen yeasts for enhanced aroma. There's sort of one famous example of hazy IPAs over here. And one of the guys from our team, Chris Saunders, he actually did a little side project pulling out yeast from that beer and comparing it to some common dry yeasts, and did find that there was a wheat beer yeast in that mixed culture. So for whatever reason, that brewery is using a wheat beer yeast. And we've also heard other people say that wheat beer yeasts can help to enhance biotransformation or enhance aroma, whether that's ester production or whether that's terpene biotransformation. So interesting stuff there, but not a whole lot of real useful information to the brewer. So what do? We do an experiment and we try to measure it and try to create some answers for you guys. So what I'm going to show you is actually a part that I held back from the presentation that I gave on Monday, where we talked about this massive split batch experiment where we tested a bunch of our yeasts against each other and measured flavor production and fermentation rate and a number of other parameters. We also were able to measure terpene production, terpene conversion in the yeasts. And so we'll present that here. So the one thing we could measure here was beta-citronellol production. And the reason this is important is because there was no measurable beta-citronellol in the wort. So any amount that we see in the finished beers from this experiment, where it was a pretty standard wort, like 35 IBU, a little bit of flame out hops, nothing, this is not an IPA, this is nothing extreme. Anything we see there is something that's being produced by the yeast from the hops. So looking at trends with the yeast groups, we see, anyone who was in here on Monday, remembers lagers are low. We love lagers because they don't really do anything flavor-wise. They leave it very neutral. But then we see some other things jump out. We see some of the individuals in the wild group, in the saison group, and in the Weizen group or German group with a pretty high beta-citronellol production. So that's kind of cool, right? That suggests to us, and especially just seeing this kind of distribution, suggests to us that we may want to look over here if we really want to be getting more value and more flavor intensity out of the hops that we're using. So who's doing that? By far, the highest producers are a lot of these saison, Weizen, wild yeasts. So we see our farm stand saison, Überweizen, and Wild Thing more or less leading the pack here in terms of the production of beta-citronellol. And even a fairly generic wort, this is still a reasonable or a lot of beta-citronellol in some cases. I did draw a little line here to show the sensory perception threshold, to show that a lot of the yeasts are above or around that value, and then some of them are quite a bit higher than that threshold value. So this is pretty flavor relevant to us. Given that most people who are looking for biotransformation are typically trying to make IPAs and using the yeast that'll make IPAs, I think it was important to highlight the non-phenolic beta-citronellol producers, because it's really hard to make an IPA with a saison yeast. I'm not going to say it's impossible. I love saisons with a lot of aroma hops, but it's a different product, right? You really do need like a non-phenolic strain to get that balanced IPA flavor and not clash with the hops in a lot of cases. So in terms of our highest non-phenolic producers, we saw the Cerberus strain, which we saw on Monday, also had a lot of interesting traits just in terms of aromas all over the place. We also saw the Hornindal Kvike punching above its weight, as well as the Vermont Ale strain. And these are all strains that I have used in the past for IPAs and hazy IPAs, and that we think do work really well in IPAs. It's not to say that any strains that are lower beta-citronellol producers are bad for IPAs by any means. For example, Foggy London Ale is very much in the middle of the pack here, but this is important because it means that different yeasts have different traits when it comes to terpene conversion, and depending on the hops that you're using, you may select a different yeast for your outcome. Another thing to mention here is that there's, these are terpenes in isolation, right? What happens when we stack that with other aroma compounds? Because we know, in a lot of cases with these aromas from the yeast, that there's additive effects. You add one thing to another thing and you get three instead of two. And there is some evidence for yeast esters enhancing or changing the perception of these terpenes. So one thing that came up in a recent study was this thing called isobutyl isobutyrate. It's a sort of pineapple-y tropical fruit ester. When that was in a beer alongside the monoterpene alcohols, it seemed to have a synergistic effect and enhance the aroma, the fruity aroma perception of that beer. So that's really cool. And I think that needs to be explored further. Of course, it gets really challenging when we're going beyond just like spiking one thing in a beer and measuring what it tastes like. When we start adding a bunch of things in there, it gets a little bit more complicated. But I'm excited about this stuff because, maybe beta-citronellol plus one yeast ester might end up smelling different from that plus a different yeast ester, and we can get all sorts of complexity that way through the yeast and hop selections. So I can't talk about the terpenes without talking about glycosides. Usually this is the one concept that people do know about when it comes to biotransformation. And that's because some terpenes are bound by these glycosidic bonds. I tried to make this slide simpler. This is about as simple as we could get in this case. So what that means is that you have basically a sugar that's bound to that terpene. And when it's in that state, it's not aroma active. It doesn't really have any flavor activity. But yeast have these beta-glucosidase enzymes that can break apart those bonds and release the terpene and make that flavor active. That being said, there's a lot of conflicting research on this topic, where some researchers have found that even though this is possible, there's yeasts with high beta-glucosidase activity and hops that have a high degree of glycosidically bound terpenes or aroma compounds. For whatever reason, the actual glycoside concentration in wort is often low, and some of the scientific results have been inconsistent or confusing. So it's still unclear how much yeast beta-glucosidase impacts hoppy beer aroma compared to just straight up terpene conversion. Something that should be mentioned here, though, is that pH does impact the release of glycosides. Glycosides can also be broken apart just by acid. Through acid hydrolysis. And so since we know from Monday that some of the yeasts drop the pH more than others, it is possible that some of the yeasts may still impact glycosidic bonds, even regardless of beta-glucosidase activity. So there's still some complexity there, and I'm not sure if that has been explored fully in the science yet. So we get into some fun stuff here with the thiols. I hadn't ever heard about hop thiols before 2015 or so. The reason being that the science on this stuff was very, very new. It's well understood in wine, or at least more cutting edge in wine, but the beer world really took a while to start to figure this out, that, beyond the terpenes, that there are other interesting aroma active compounds coming from the hops that may explain why people are paying so much for Citra. So what are thiols? Again, there's a lot of different ones, just like with the terpenes, but we're going to really focus in here on 3 characters. So we're going to focus in on this one, 4-MMP, which has a blackcurrant kind of aroma. And if you've smelled blackcurrant, it's a pretty distinct aroma. if you hear people talking about Sauvignon Blanc, this is really the key aroma compound, and you may also hear people call this like cat pee. And again, if you hear the term cat pee, that may also make you think about citra hops, which certainly do have an appreciable amount of this 4-MMP in them. So it's kind of a weird smell on its own, but when you combine it with other fruity compounds, it can really enhance that perception of aroma and intensity. We're also going to talk about 3-MH. This one has a very grapefruity aroma to me. Some people say rhubarb as well. Just really, really sort of fresh aroma. And then also 3-MHA, which has a very tropical fruit kind of character, like guava or passion fruit. Both guava and passion fruit have similar aromas to this. And the commonality with all of these, and I think I hinted at that a little bit earlier, is that they all have sulfur in them. You can see this SH group in all of them. And for whatever reason, to our noses, that SH is like, it's like a siren. It's super loud. It's super noticeable. You never mistake, the nose never mistakes SH. So for that reason, these things are super flavor active at really low concentrations. If we look at those concentrations, those are nanograms per liter. So less than 1,000th of the amount of the threshold for some of the esters that we were talking about on Monday. They can be present in really tiny amounts and still be very flavor active. Unfortunately, in a lot of cases, they're also present in tiny amounts in our precursors, like in our hops as well. So it sort of goes both ways there. What hop varieties do you find thiols in? And the answer that I give is, if it's expensive, it's probably got a lot of thiols. This information is surprisingly and frustratingly hard to put together. So I had to put together some of this information from the couple of scientific papers that are out there, but then also conference presentations from the last 3 or 4 years. I'm very fortunate that I get a chance to go to a lot of the brewing science conferences and collect and soak in all of this information. But I was finding, especially when we talk about thiols and hops, it's really, really inaccessible and hard to find. Even this list of 5 hops was cobbled together from 2 different studies. But yeah, if the hop's expensive, it's probably got a lot of thiols. These things are super aroma active. They smell great. They mean that the hop has a really strong aroma impact. So if it costs a lot of money, it probably has a lot of these things in it. So Nelson Sauvin, Citra, Mosaic, Amarillo, Mandarina, these are all, classic, very strong aroma hops that are also classically expensive. So hops have these thiols in them, but what is yeast doing here? And the answer is that many of the thiols that are found in the hop material, not all, but many, are bound they're bound by a cysteine group. So they're bound up, just like with those bound up terpenes, they're bound and not flavor active. But luckily, our yeast have enzymes that can break those bonds. So just like the beta-glucosidases for breaking off those terpenes from the sugars, the yeast has a cysteine beta lyase that can break that apart, release the thiol. The yeast can use the cysteine for energy. We talked about back in Yeast Basics how yeast needs amino acids to make more proteins, and cysteine is one of those. So they're able to do this, get a little bit more building blocks, a little bit more amino acid Legos to play with, and then release something that's tasty to us as well. So it's kind of a nice mutual thing that we've got going on. So we can dive in a little bit deeper, just because I think it's kind of cool. What's the yeast doing? And I just sort of mentioned this briefly, that there's this beta lyase enzyme called IRC7 that is really in control of this activity. So one thing that has been studied, and luckily this is a really important problem over in the wine world, because they want to have these really expressive Sauvignon Blancs and other types of aroma active white wines. They've found that depending on the type of IRC7 gene the yeast has, it can influence the activity of the thiol release. So, and that a lot of the yeast have a deleted or a deactivated form of this gene. So if you look at the chart on the right, you can see the one on the very left has the short version of the gene. The one in the middle has one copy of the short version, one copy of the long version, still less active in release of 3MH. And the one on the right-hand side has fully intact IRC7, and you can see that it's also releasing more of the 3MH in the wine. So it seems to have some control over this aroma production. In general, it seems like a lot of the domesticated yeast have these shorter gene mutations. And so it may mean that some of our highly domesticated beer yeasts aren't very good at this reaction, although it's still, not very well studied. But it does seem like wilder yeasts are more likely to have this intact. And one thing that's known is that a lot of the non-Saccharomyces cerevisiae yeasts, so things like Saccharomyces bianus and uverum, those are more common in winemaking. Those often have really, really high thiol release activity, and that may have to do with having beneficial or no mutations in the IRC7. This is just starting to be studied in beer. So this is from a paper that came out just in 2019, where they test. This is from Technical University Munich, Weinstefan. They tested 6 of their yeast strains for release of 3MH and 4-MMP in some beer, and unfortunately, it's hard to find any kind of conclusive results here, where they actually didn't see any statistical difference between the yeast with that grapefruit aroma, but they did with the black currant aroma. So there's some indication of yeast control of thiols in beer, but it's still very, very cutting edge and hasn't really been explored very well yet by scientists. But I'm hoping that more of that happens. One of the challenges is that until recently, it's really only been one lab in Japan and one lab in France studying this stuff. But now it seems like more people like these guys in Germany are starting to study it. So with more competition, we usually see results a little faster. And given that the most expensive hops seem to have a lot of these aromas in them, there's definitely an economic incentive to understand how to actually get more of these things out of the hops and into the beer and keep them in the beer. So that was a lot of science. I hope you're still with me. Now that we've absorbed that information, we can actually get into some practical tips. We really needed all of that so that we can start to form some ideas for how we can be a little bit more strategic about designing beers that leverage biotransformation and biotransformation properties. So when you're selecting hops, you can select hops for higher concentrations of these biotransformable chemicals. So again, this is I had to go and pull data from a bunch of different conference presentations to put this together, but it does seem like there's a few different hop strains that come up pretty regularly as that have heavy terpene content, and that's Bravo, Centennial, Cascade, Chinook, Mosaic. Some of those are not hops that you would ordinarily put into a New England IPA, right? I get the feeling that hops like Bravo and Centennial and Chinook are kind of out of fashion now, but the fact remains that these may have some utility in these types of beers because of the flavor precursors, and that if you're pairing these hops with a yeast that's able to release that flavor, there actually may be some very beneficial things here, especially when you consider that, with exception to Mosaic, most of these hops are pretty affordable. When we look at the thiols, again, we have the list of hops that we have been able to dig up studies that show high concentrations. So it's all the usual suspects. One thing to mention is that Cascade does come up more than once as being a hop that does have appreciable amounts of thiols. And given that Cascade is often, I don't know, half the price of Citra, there might be some biotransformation potential, aroma potential there. So don't overlook Cascade just because it's old school. And in many ways, it still punches above its weight. So we can also select our yeasts to maximize these biotransformations and maximize these aromas. So if we're trying to get more terpene aroma, terpene biotransformation from our yeasts, we can definitely select some of those high converting phenolic yeast. So like the Uberweizen or that farm stand saison, or most of the saisons, or the wild thing. These are all really good at the phenol conversions. I've definitely made a number of beers that contain lots of aromatic late addition hops and these types of yeasts, and you can get really cool results if you're making like a fresher, hoppier saison, for example. I think more relevant to people because of just the sheer amount of New England IPAs and IPAs in general that are being made is the clean ale strains that are not phenol producers. And what we saw in our study was that the Cerberus strain, the Vermont ale, and the Horndal Kvike were really punching above their weight in terms of terpene bioconversion. So those could be used on their own or blended with something else to try to get more of that type of aroma out. In terms of the thiols, again, like a lot of the research is just so new and so cutting edge. There's just really not a lot of information out there. I will say that we're working toward testing this in our yeasts soon because we think that it's really important, right? If we can help brewers to use less hops and spend less money on hops, then I view that as a good thing because it helps with the bottom line of those brewers. So we really want to understand this in our yeast to help brewers maximize flavors from the yeast and the hops. One thing I will say is that many wine yeasts are already marketed for thiol release. So you can just search like wine yeast thiol kind of thing into Google and you might get some search results. And you can use those in beers. You can use those as a co-pitch with a yeast that can ferment all of the malt sugars. But make sure that yeast, if you're selecting a wine yeast for thiol release, make sure that it's a non-killer yeast. And that's one of the challenges because a lot of the wine yeasts have this killer factor that helps them compete with non-killer yeasts and kill them. The other thing to mention is non-Saccharomyces cerevisiae yeast within the Saccharomyces genus, like Bianus and Uverum. If you get your hands on some of those interesting species, they have a lot of biotransformation capacity, and they seem to have some thiol release capacity as well. And I can say, we have this Bianus strain that we grow for a client, and if we grow it in hopped wort, it's always like crazy aromatic. So there's definitely some potential there. Timing is really important for this stuff too. So, you can choose the right yeast, you can choose the right hops, but if depending on how you choose the timing, you may still not be using these things very efficiently or getting the most punch out of them. So for terpenes, it seems to be typically recommended that these can be extracted pretty efficiently through late kettle or whirlpool additions. Those glycosidic bonds can be broken by heat as well. So that's a good way to just get those things into solution potentially and be able to be converted by the yeast. And then also early dry hopping can also help with the terpenes. Also just to have contact with the yeast and make those chemical reactions happen. In terms of the thiols, while they may, it's possible they could be more soluble with late kettle whirlpool additions versus dry hopping. Because they're so volatile and present in such tiny amounts and sensitive to oxygen, we do typically recommend that those thiol forward hops are added during dry hopping, at late ferment or post ferment. Really, the closer to the packaging, the better, simply because it's, you can do all of this work up front to maximize the thiols that are in your beer and get that, awesome tropical fruit, grapefruit aroma. But then, the tiniest bit of oxygen can take it all away again. So you're really sort of fighting the battle on both sides with those guys. So closer to packaging seems to be better. So just to provide an example, just from a beer that I brewed back in the winter, leveraging some of this information and really trying to get a nice tasting beer that didn't use a ridiculous quantity of hops. The goal here was to make something that's really hoppy and tasty, but 3% alcohol. So I made this beer, and in terms of the hops, I included Bravo and Simcoe in the whirlpool. And the reason being that Bravo is really high in geraniol, which can be converted into the beta-citronellol and give you that nice lemongrass aroma. Simcoe is also really high in 4-MMP, that kind of blackcurrant, cat pee kind of aroma that really supports and enhances some of the other fruity characteristics in the beer. So getting those in early seems to be beneficial. And then in the case of this beer is fermented with a blend of our Cerberus and Vermont ale strains. The reason being that we saw the Cerberus and Vermont were both quite good at the terpene biotransformation. The Cerberus is also quite a good ester producer, but the Vermont ale ferments a little faster and drops out, which can help with the astringency of the beer. And then dry hopping the beer with hops that have high thiol content. So in this case, Citra and Simcoe. We know Citra it has these thiols in spades, and that's part of why Citra is so expensive. And adding those at the end of ferment, within 1 Plato of final gravity. So we're not oxidizing the hops. There's a little bit of fermentation going on. But if you were a pro brewer, you could harvest some of your yeast at this point and still dry hop and still get a pretty big dry hop impact. So that is something worth trying for those who are dry hopping super early. Just try it later on. In this case, it was a short contact time too. There's not much evidence supporting contact times of more than 3 days for dry hops. So sticking with that to avoid any harsh extraction of any of the crazy bitter or astringent things from the hops. And I think that beer turned out pretty well, and really nice to be able to use some of the data that we produce at the lab to make decisions about the kinds of hops that I select and the kinds of yeast that I select for these types of beers, so that I don't have to just dump tons and tons of money into a fermenter, and I can be a little bit more strategic. This used about half as much hops as I would normally use in a New England style beer, and I think tasted almost the same. So obviously we need to do more experiments on this and collect more numbers, but I think there's a lot of potential there. I think a lot of people want to know what these things taste like. And I think that's really important to know what hop aromas actually smell like, as opposed to just the hop on its own. You have so many different things there, so many different aromas. It is nice to sort of break it down into its individual components. So one thing I spotted recently was that the hop company Barth Haas, they have a hop aroma kit. So I know that's available online. If you just search Barth Haas hop aroma kit, you can purchase that, and they've got these aroma standards for different traits of the hops, like citrusy and floral and things like that. And I imagine that those are blends of some of these aroma compounds that we've just talked about. One other thing you can do is, as we talked about, a lot of plant essential oils are really, really rich in these terpenes and thiols. And so you can also purchase plant essential oils. I've got a lavender essential oil that's very rich in linalool. Lemongrass would have a lot of that citronellol in it. And then a grapefruit essential oil should have a lot of the 3-MH as well, because that 3-MH grapefruity sulfur note is pretty characteristic of grapefruit itself. So that's a cheap way to sort of buff up your sensory knowledge, and maybe you have these lying around the house already, and it's another way to do something with these random bottles of essential oils. More learning. I've really just scraped the surface of this stuff. It gets really deep and really complicated. Had to jump between a lot of different scientific articles, conference presentations, to put together the information that I presented today. And it's still very much surface level. If you do want to dive deeper, I would recommend Scott Janish's book, The New IPA. He had a blog previous to this and did a lot of really cool deep dives into just certain parts of IPA production or hop aroma. And the book sort of takes that a step deeper. And really covers all of the science that's out there on this stuff. So if you want to go deeper, get a little nerdier, then that's really the way to go right now. So in conclusion, we can use what we know, right? All of the science that's out there, we can use that to enhance the hop flavors in our beer. We don't have to guess. The right mix of hops, yeast, and timing is important, but it's still being studied. There's still a lot to learn. Biotransformation is complicated, but there's a lot of smart scientists working on it. And that's really exciting. It seems like there's something new and interesting every few months out there, and especially given the economic value of hoppy beers now, I expect that will continue to be the case. And we're going to continue to learn more about this stuff and be able to use this information to make beers that are tastier and less cost intensive. What's next? At least on our side, Escarpment Labs is committed to learning more about biotransformation capabilities of our strains, especially when it comes to the thiols. The reason being that we want to help out our customers, and if there's a way that breweries can spend less money on hops, then that helps their bottom line, and that's a good thing for us at the end of the day, because now more than ever, the industry is facing a lot of pressure to stay afloat and tighten up margins. And so this may be one of the paths forward, is being smart about leveraging the science to make the beers and the businesses as viable as possible. So that concludes this presentation. Just wanted to flag that I don't brew a lot of hazies. I don't always brew hazies, but when I do, I use science. And I'm still learning, but being able to do this stuff has avoided a lot of trial and error, which is really exciting to me. And one of my goals is to help people avoid having to make too many mistakes. It's good to make some mistakes, but there's so much knowledge out there that can help us to avoid a lot of the easy mistakes. So hopefully that was helpful. And then if you have any questions here, you can ask it in the Crowdcast platform. And then you can also email me, just Richard at Escarpment Labs. I got a few emails from the last talk, and I try to do my best to respond to those in a reasonable time frame. So if you do have any, especially like the more in-depth or specific questions, just shoot me an email, and I'll do my best to get back to you. So I'm going to invite Nate back in here. One of the quirks of this system.
I figured it out. Great. That was wicked. Hello. That was wicked. Thanks, Richard. There are a handful of questions that I want us to kind of go through. And I want us to kind of touch on a few of them. There's a few that are a little kind of, I don't think they're exactly knowable. I don't know if we can actually answer some of these questions, but we can kind of give our best crack at them. But we'll start from the top of the most voted. So the first question is, usually when I hear about biotransformation, is the relationship between yeast and hops. Is there biotransformation between yeast and other compounds from malts, spices, and fruits, etc.?
Absolutely.
Yeah, absolutely. My immediate thought on this is that you see this with wit beers. Coriander is really high in granulolitol. So if you actually want to add more citrus tones to a lot of wit beers, you have to add coriander, which is always kind of it's not how you think, because you're already adding bitter orange peel. So you think it would be present, but it's actually the opposite of what you think.
And maybe use a Weizen yeast instead. That's my my controversial tip.
Or a Trappist yeast.
Yeah.
The Ardennes makes great wits. We don't understand it, but it's wonderful.
Mm-hmm. Mm-hmm. And I would say even with fruits, like that's one case where the glycosides may really matter. There's some work that was done on glycoside activity with Brettanomyces in like lambics, fruited lambics, and it seemed pretty clear that it was able to release some of these bound flavor precursors from things like cherries. So very much, if you're adding fruit or spices to the beer, you can expect some kind of, if they've got glycosidically bound aromas or terpenes or thiols or whatever in them, you can expect the yeast to do something.
I do want to put one thing out, because I see this in the comments, and I'm glad Dave Henry just brought this up. You don't have to whirlpool or hot side a lot of those spices and herbs. If you do, you will get glycoside breakdown, which you won't get. Thermal glycoside degradation, which you won't get on the cold side, because obviously there's no heat. But you can add coriander in wit beers with great success, either in fermentation or as you would a dry hop. That's a really useful way to kind of add those flavors to it in a bit more of a delicate way. We'll go to the next question because this one kind of I thought was kind of interesting. Recently used a Kvike strain, not Hornindal, unfortunately, for several hop-forward beers. They all showed a surprising lack of dry hop aroma. Process rates per barrel weren't changed from previous batches, just swapped the yeast from more expressive Belgian or English strains. Any theories as to why dry hop aroma would be reduced with a cleaner yeast? They're probably not going through biotransformation in either ester, with either sulfur compounds or all the essential oils we talked about today.
Yeah, one thing we saw with the Kvike group is that there was like quite a lot of variation with the terpene conversion. So that's possibly the case, is that whatever that person used may not have as much capacity for the terpene conversion. The other side of that is that there are some hints that the yeast pitch rate and temperature can impact, at least on the thiol side, can impact the release of those compounds. So like in general, it seems to recommended lower pitch rates. And that might be it with the Kvike too, is try a lower pitch rate. And that is something we did see, at least with a couple of the Kvikes when we did that pitch rate study, was with a lower pitch rate, we saw a little bit more of the beta-citronellol. And that was actually true of the Vermont strain as well. So there is some room to play around with pitch rates for enhancing this as well. But there's a trade-off too, because underpitching the yeast can also cause other flavor changes.
Yep. Fusels and other negatives that you don't necessarily want in that beer.
Yeah.
This one was kind of fun. When co-pitching yeast strains, back this up a little bit. Would co-pitching yeast strains, which produce the necessary enzymes to liberate terpenes/thiols alongside a primary strain, promote these flavors? Like just enough for them to output these enzymes without leaving themselves contributing much flavor. That's kind of the idea behind Julius.
I think that's what Treehouse is probably doing. Yeah. I would love to just have all of those traits in one yeast, and that's what we're working towards behind the scenes. But that seems to be the idea there, is just get the enzyme activity from the Weizen yeast. Yeah. I guess. I don't know.
Theoretically, if the Weizen yeast would, they may not be that expressive, based on what sugars it's fermenting and what its pitch rate is across the entire board.
Yeah.
If you're worried about the phenol portion of the Weizen yeast coming too forward, I would probably just focus on making sure you don't have a lot of wheat, oats, rye, things like that inside there. Stay well away from doing like a ferulic acid rest. Like there's probably ways to make this beer, make the phenols in that beer not be flavor forward.
Yeah.
This is a quick one. Does temperature affect biotransformation? We didn't really talk about that at all.
Yeah, didn't really get a chance to get into that, but in general, in general, like the closer you are to sort of the optimum temperature for the yeast, the better. Like there's very limited data with the thiols, but it did seem like hotter temperatures was a little bit better for thiol release, and I believe the same for the terpene release as well, just because. I'm guessing at those higher temperatures, the yeast is just more metabolically active as well. That being said, I wouldn't push them like crazy hot in an attempt to get more biotransformation, because you might flash off other flavors, unless you're doing like Kvike and you can just get it hot and done super fast.
The kind of follow-up question was, what about dry hop at Kvike temperatures?
I wouldn't do that. Yeah,
I wouldn't either. My understanding is that thiols are also much more they're much more delicate. They're more likely to flash off.
And that's sort of why I made the recommendation, is if you're trying to preserve thiols in the beer, it might be a good idea to dry hop closer to the end of the ferment. If you're doing the mid ferment dry hop with a Kvike at high temperatures, you're probably going to flash off a lot of aroma. Like you're going to have a lot of money coming out of the tank or fermenter. Yep. Just a guess. I don't have measurements on that. And the other part of that is that it's, again, I don't have measurements on this, but I suspect it to be the case that some of the like unpleasant hop things are probably extracted at better efficiency at higher temperatures too, like the polyphenols that give the astringency, the hop bite. guessing those are probably extracted better hotter too. So you may want to avoid that. Like with Kvike, what I've done is get it through the primary ferment and then let it gently crash to a normal temperature and then dry hop. And then even if there is some hop creep, the yeast is still active at 18 to 20 Celsius and it'll still get through it.
Yeah, especially because it's likely glucose with hop creep. I just want to do a quick follow-up on this. So someone asked, what about high Kvike temps under pressure? I think the negative there is that you'd still be getting the polyphenols and tannins that are associated with hop bite. So it probably, you'd retain some of the flavor compounds, but you'd also be extracting compounds you don't want. So
yeah, that's the risk. And like that's something that I've seen from I made a saison once with it was like Nelson at the Whirlpool, and then it was like saison but with Kvike yeast as well. It was blended saison and Kvike yeast. Nelson in the Whirlpool, and then dry hopped it really early, and while it was still warm, and it was like undrinkably bitter and astringent, and like ended up dumping it. So that's one anecdote, but it's challenging.
This next one took me a little bit by surprise, and I've heard of something like this beforehand, but it was a little bit weird. This is from Jason Crossett. Would oxidizing dry hops a bit in the cooler to increase beta acids help increase the rate of biotransformation occurring in the beer? I already answered this. I don't think so, mainly because you'd be promoting oxidation of beta acids, which are about 80% as bitter as isoalpha acids. And you'd be oxidizing a lot of the sulfur compounds. The enemy of thiols or just sulfur chemistry in general is oxygen. So I probably wouldn't want to recommend you doing this. But I wanted to get your thoughts on this because this seems like it's something that I would not recommend anyone do.
What's the I'm not sure if I understand the concept.
So the idea is that by oxidizing your hops a little bit, I think you oxidize your hops a little bit, you get more biotransformation. It doesn't seem, it seems kind of things like these two things are completely disjointed from each other.
Yeah, no, that very well could be a thing. I know that this, especially on the terpene chemistry side, there's tons and tons of research out there. So it could be a thing. I actually don't know.
Okay. I've never come across anything like that in my readings of this.
Oxidized beta acids just makes me think of like aged hops for lambic and stuff, where you want the antimicrobial activity, but not as much bitterness. So maybe that's the idea, is reducing bitterness. Maybe, yeah.
This is a question from Germany, which is kind of cool. Have you investigated this is technical, so we're going to lean on you on this one. Have you investigated the release of hop volatile thiols from precursors during fermentation through yeast beta lyse? What else yeast strain enzymes could be taken into consideration for increasing 4-MMP and other thiols and aromatics?
So, it's not just IRC7. There are other genes involved in that activity, and that has been somewhat explored. There's still definitely room to improve. And clearly just looking at some of those results from just the IRC7 mutations, and given how much variance there is, there's probably other things underlying that as well. So I'm not providing a very clear answer here, but the answer is that it's not as simple as I explained it. There are other underlying genetics and enzyme activities that we didn't get into. And if you shoot me an email, I can follow up with some papers.
All right. A few just little bits, and I think we have time for about 2 or 3 more. Sure. There's apparently a part in the new IPA about oxidized hops, so I think I'm gonna address that and see where that takes me.
Okay.
Is there any benefit for having the same or similar essential oils and or terpenes thiols from different sources? So kind of the idea of trying to generate linalool and geraniol from coriander and from hops at the same time.
There could be synergistic effects. I think it was Mike Tonsmeyer who did some like homebrew experiments with hops and coriander, and yeah, yeasts that were allegedly biotransformation active. And I actually don't recall the outcome of that, but I know that has been explored. Certainly, like some of those things, like I bet putting a little lemongrass into an IPA with some of those traits would be really nice, and that could help support it. And in the case where you have all of these different terpenes being converted in all these different ways, then straight up adding in the thing that smells like that terpene could sort of drive the profile in one way, right? If you really want it to have that lemongrass, beta-citronellol kind of character, then yeah, put in some lemongrass. It's probably cheaper than the hops.
It definitely is cheaper than the hops. I think there's a 2 or 3 more that I think we're going to go through. Just apparently the oxidized terpene thing. Apparently, oxidized terpenes are more soluble. It's part of the reason why.
Okay. So that would be pretty relevant when you're talking like whirlpool hopping, then, or potentially relevant when you're talking whirlpool hopping.
Yeah.
Is that oxidized terpenes or oxidized sesquiterpenes? Because I mean, I know that these are different things.
Yeah.
Just want to make sure that that's not being misconstrued.
I'm going to suggest that we read and consult that so we can get a proper answer on it.
Okay. Okay. I know about those ones. I know what's going on there because that's some logger stuff. But.
Yeah. The next question I think is a little bit. I think we need to kind of clarify what the scenario is. Would it be better to try and blend hops that are terpene heavy aroma and ones that are thiol heavy, essentially, and not mix? Like, should we mix them or should we try to focus on one or the other?
You can mix them. Totally. I think it's like if you go back and just look at the the hop blends and recipes of what I would say the big names in these kinds of beers are, you often see already blends of these things. Like you go and you look at some of these guys and it's like, okay, why are they always using Centennial? Why are they always using Columbus? Like what the heck's going on here? And like I imagine it's to get a balance of these things, because like if you're it's like if you're just using Citra, it's like you're a band with no bass player kind of thing, right?
You're Metallica.
You kind of need some of those foundational notes in there, right?
Yeah. I would say this though, that when you it's Richard said this earlier, but when you have the two compounds present, it really is the beer is greater than the sum of the parts. Like if you have thiols plus terpenes, the sum of those two is going to taste more intense than the individual components on their own, and it may also taste unlike the two individual components on their own. Like you see the way in which the human palate detects these things shift quite heavily.
Mm-hmm.
Well, 2 or 3 more. Could you explain a bit about hop dosing rates for hazy and NEPAs? I think we're going to talk more about that when we talk to Barn Cat. So I'm going to kind of park that question for now.
Yeah, it's all over the place, and a lot of like the amount that you need depends on some of the stuff we talked about today, but then also just how much are you able to extract from the hops, how much are you able to avoid the oxygen, all of the. There's so many variables, man. Yeah.
Second last one, when co-pitching yeasts, what can be harvested to reuse? Yeah, great question.
Yeah, in general, what we've seen with yeast blending or co-pitching is that if they have a similar growth rate and attenuation, they typically can stay in harmony, so to speak. If the growth rates are mismatched or if the attenuation is mismatched, then that can cause one to outcompete the other. So for example, like the kvikes that we blend, they stay quite stable. Like we've gone out to 5 generations at least, and seen them stay stable. But then if you've got 2 yeasts that have mismatched growth rates, then it's going to go out of whack after a generation or 2.
Yep. And the final question here, and I thought this was kind of interesting, and this is, again, I suspect this might be slightly unknowable. Do we know if whether or not hop extracts and other kind of hop products will react differently for biotransformation and terpene content and things like that? My gut would say no. that they might be lower in thiols, but that's my gut instinct. I don't have much to back that up.
Yeah, I think getting first of all, like making a thiol extract from these things would be really expensive. And like even the fruits, I mean, well, grapefruit's one thing, but like even if you're trying to get an aroma extract from like guava, it's still going to be expensive to make.
Yes,
for some of these things, for sure. Like I know that the hop companies are getting more and more, technical about the products and fractions that they produce, and as our sister industry gets a lot smarter about figuring out how to fractionate all sorts of different terpenes for medicinal and recreational use. A lot of that technology is translatable to our field as well. There also, it may be a very near future where there's a huge amount of waste terpenes being produced by the cannabis industry that may be used in beer for aroma. I very well expect that to come down the line in the next little while.
Yep, that makes sense. I just to kind of summarize this, and I'm fairly certain I have this straight. Sulfur compounds are much more volatile than other flavor compounds. So you'd probably assume that when you're making a product or making an extract, that they would be they wouldn't be as present. They probably wouldn't be as dominant, and then they wouldn't be in the raw material. I know you see some people make arguments for this when it comes to using whole cone hops versus using pelletized hops, especially with the cryo hops. It's one of the things that people kind of push back on a little bit, is because you're going through more processing on these things, which is causing some unintended volatilization to occur. Like the extrusion process for taking hops, like the shredding and then extruding, is an incredibly high heat, a high friction environment. So the thought here is that you're going to be losing some of these more volatile compounds. And like there's been a huge increase in the technology associated with hop drying in the last couple of years. Like if you were to go back 20, 30 years, you'd be using high temperature kilns, no nitrogen purging, it'd just be blast everything off and you're left with, your noble aromatics. Something that's been kind of, I've had a few beers the last couple of years with things like Columbus and Chinook, and there's been a few where I think you mentioned Columbus earlier, but how I remember these hops tasting.
Yeah, no, Chinook has changed radically.
Yeah, almost a decade ago.
Radically.
Yeah, they don't taste the way they used to do. The way the increases in processing really have helped preserve a lot of these previously consistently lost flavor compounds. So you're going to see differences in these hops. If you haven't brewed with them in a while, I'd really recommend you revisit them. Just because, YCH has really stepped up its game for handling these things. They really have. Same as Hopsteiner and all the different, the German producers as well. I can't say a bunch about the UK producers just because I honestly don't use a lot of them. But hop processing is a big thing. It's a big impact. I think that's a good place for us to wrap up. There's one or two questions in there, but I think they are kind of best either suited for a Q&A or for individual responses. I just want to remind everyone again, so we do have more talks next week. We have the Road to Lacto 2.0 next Tuesday, and then we have a talk with Barncat Ales a week today. And then the following week, we have our webinar series. And then any additional talks we'll be announcing after that as well. I think that's a good place to end it. Anything else to wrap it up with?
All good. Yeah. Looking forward to all the future webinars. Looking forward to taking a couple weeks off from presenting and getting back into it with some special guests. We've got Lars Marius Garshall and a few other people joining us on the week of April 20th. Yep. It's going to be good. All right. All right, everyone. All right. Have a good evening. Thanks, everyone. Happy long weekend and Easter and things like that. And hope you guys stay safe. Cheers. Cheers.